For our final project, our group analyzed the use of fuzzy clustering with biomedical data. In this presentation, I will go over the general background knowledge that pertains to our project, the significance behind our work, what fuzzy clustering is and why we are using it, the methodologies that we utilized, how we processed our input data, and the algorithms we used. I'll first start with some basic background knowledge. Cells are the basic building blocks of the human body, and the human body is composed of approximately 37.2 trillion cells. Each of those cells contains genes and the proteins required for the cell to carry out their designated task inside the human body. It is normal for the cell to divide, which can cause proteins to behave abnormally and the cell to go out of control, which occasionally results in the creation of cancerous cells. So what are we doing? Biological data cannot be processed by people alone as it is too large, so we need computational methods to analyze this data. We utilize different computational methods to group the genes with similar functions into clusters and then analyze the results. This can be used to infer the functionality of novel genes by grouping them with genes of well-known functionalities. So what is the significance behind our work? Lung cancer is the leading cause of death among cancer patients worldwide. By using fuzzy clustering, we are able to simulate real biological problems and gain crucial insight into the genotypic and phenotypic factors that cause cancer development and will help further the advancement of medicine. So what is fuzzy clustering? Fuzzy clustering is a clustering method used when each data point can potentially be part of multiple clusters. Each data point is assigned a membership value between 0 and 1 for each cluster, indicating the degree to which the point belongs in said cluster. The closer the number is to 1, the more the data point is in that cluster. There are many different forms of fuzzy clustering, 3 of which will be described later. Using fuzzy clustering with biological data allows us to account for any uncertainty that exists in the biological processes, as gene products contain multiple functional domains and can affect more than one biological function, either indirectly or directly. For example, one gene could be a part of both the metabolic pathway and the immune system pathway. We use this method to assign a membership value to each gene in regards to its relation to lung cancer in order to help us determine which genes are more prevalent than others in lung cancer. This brings us to the methodologies that we utilized. As stated earlier, we used 3 different fuzzy clustering algorithms to analyze differentially expressed genes in lung cancer patients. This helps us understand the subgroups that share genetic expression profiles. We used the lung cancer genotype and phenotype data from the GEO database. The genotypic data was then filtered and organized, keeping only the genes that are most relevant to the disease development by finding the differentially expressed genes. This was done by using a Java program as well as R script and the EdgeR package. In order to use fuzzy clustering, we first had to determine how many clusters were needed. This was done using the elbow method, which is designed to find the optimal number of clusters in a dataset. From there, we use the fuzzy C-means, co-clustering, and bi-clustering algorithms to cluster the data. The fuzzy C-means algorithm uses randomly assigned coefficients for each data point to cluster the data. This is an iterative process that computes a centroid for each cluster and then uses the distance between the centroid and membership value to update the coefficients, repeating until convergence. The next algorithm we used is the Information Bottleneck Fuzzy Co-Clustering Algorithm, or IBFCC. IBFCC assigns a membership function to both the objects and features, which in our case are the patients and genes, and uses the information bottleneck theory to measure how similar the genes of each patient are to how the genes are clustered. The last algorithm we used is the multi-objective genetic fuzzy bi-clustering algorithm, which uses a multi-objective optimization fitness function to extract a bi-cluster. We then analyze the results from these 3 fuzzy clustering algorithms.